Today's session is part two of a two-part session covering the topic of common data models. And we are extremely fortunate and thankful today to have James and Judith join us today, both of whom are experts in the use of common data models. Next slide, please. So as Alex said, the recording of the first part of this two-part session is available on the HDRN site. In that session, just to recap, we provided an overview of common data models, what they are, and the organizing principles of common data models, and the two main categories of common data models: organizing, such as the Sentinel, and mapping, such as OMOP. We also provided an overview of some of the Canadian applications of our Canadian adaptation of the Sentinel Common Data Model that Judy has helped us with over the years. Today, we're pleased to have Jamie and Judy join us to talk about initially an introduction to the OMOP Common Data Model that Jamie will present, followed by a discussion by Judy of the strengths and limitations of mapping common data models, mapping to concepts, followed by a Q&A session that will be moderated by Robert. And without any further delay, I'll turn it over to Jamie.
Thanks, Michael. Hi, everyone. I'm Jamie. I'm a drug safety scientist at J&J and an almost 10-year deep collaborator with the observational health data sciences and informatics community and a doctoral student. I'm improving drug safety evidence to help patients, healthcare providers, and governments make better decisions, and I'm doing this by analyzing routinely collected healthcare data from around the world. But sources differ across the world because people, health systems, and data capture processes differ across the world too. So I'm addressing those differences in my work. And doing this will improve evidence reliability that will ultimately improve patients' lives. Next, please. So I work at Janssen Research and Development, a J&J company, but these views aren't those of my employer, but rather my own and shared by many in the OHDSI community. Next. So I'd like you all to leave this discussion today knowing that data transformed to the OMOP CDM are of high fidelity to the source data. And secondly, that CDM-enabled standardized and federated analytics produces reliable evidence on which we can confidently make decisions. Next. So the OHDSI community was founded in 2014, and it's a multi-stakeholder, interdisciplinary open science collaboration with a shared goal of deriving value from healthcare data through large-scale analytics. It's also an international data network of many databases from many countries covering a great number of people. And among things, OHDSI maintains the Observational Medical Outcomes Partnership Common Data Model, or OMOP CDM for short. Next. In addition to defining and maintaining data structure and content standards, the OHDSI conducts methods research and codifies methodological best practices into open source software. And this combination of data standards, methods research, and software development supports evidence generation that is repeatable, reproducible, replicable, generalizable, robust, and calibrated. And although it's easy to get deep enough in the weeds of data standards, methods, and software development that it can seem like these are ends in themselves, they're not. These tasks are a means to an end of consistently generating reliable evidence to inform decisions to improve patients' lives. Next, please. So this cartoon is a tongue-in-cheek representation of how early distributed analyses were conducted. So imagine the iron is a comparative cohort study design intended to estimate the risk of gastrointestinal bleeds in patients with rheumatoid arthritis around the world exposed to diclofenac relative to ibuprofen. Now say we wanted to conduct this study in databases from North America, Asia, Europe, and the UK. The plug represents how you would apply the design to disparate data sources, but finagling the plug for each data source, and by finagling the plug for each data source, You can specify the design to run against each, but the solution's not scalable. It's opaque, it's resource intensive, slow, and prohibitive to non-expert routine use. So this amounts to an individual rule system per database. Next, please. This cartoon, though, is the depiction of the 30,000-foot solution. So by this approach, you're not changing the study implementation to accommodate the database, but rather you're transforming each database to accommodate standardized software. The CDM required for this task is an organizing CDM with a mapping system designed in concert by an array of experts, including source data holders and their stakeholders, data engineers, informaticists, and clinical researchers. Next. So this, I'll spend a little time here. This is the relational database schema for the OMOP CDM. I'm explicitly here to provide facts about the OMOP CDM, how it facilitates distributed analysis, and offer an example use case. I'm less here to comment or conjecture on the merits of other data models or provide opinions about one model versus another. The OMOP CDM standardizes the structure, that is the tables and fields, to represent varying data types used for analytic use cases in the real-world evidence community, namely characterization, prediction, and effect estimation. These use cases include pharmacovigilance, but also include comparative effectiveness, precision medicine, disease natural history, quality improvement, and health technology assessment. The community of data holders that use the OMOP CDM are all interested and committed to scientific collaboration to answer common problems, but they hold a variety of data types, including claims, EHRs, national registries, disease-specific registries with survey data, and biobanks from 49 countries, each with their own data structure covering different subsets of data domains and source vocabularies. So given all of this, the intent of the OMOP CDM is to be a data structure that accommodates the source data with no information loss or alteration and facilitates network research by providing common content terminology. I'll reinforce this by saying, if you want to perform analysis across databases with different vocabularies, you need a common vocabulary, and having one standardized vocabulary allows this. To be clear, every data holder can run studies on their own data with their own structure and their own vocabulary using their own software tools. But to run any consistent federated analysis across the world, you need terminology standardization. I certainly feel more comfortable interpreting heterogeneous multinational database study results where vocabulary and implementation heterogeneity has been removed. Studies of this kind are certainly easier to design and execute with a single common protocol and a single analytic implementation. Next. Having said all that, a standard structure and a standard vocabulary are insufficient alone to conduct international network studies. A rigorous extract, transfer, load, or ETL process additionally needs standardized conventions for how and which source data will be mapped, and 2, a standardized approach for data quality assessment to determine if the data are fit for use. The European Health Data and Evidence Network is a clear example of database heterogeneity that relies strongly on local data experts to drive the ETL process, indeed making the 187 CDMs in this network each a site-specific adaptive rule system. And very importantly, the OHDSI standardized vocabularies are an evolving set of dynamic content and conventions that evolve over time with community experience and input, meaning that the details of a specific code mapping many years ago might not apply to the current vocabulary. But don't let vocabulary mapping detract you from the main message. The discussion isn't just about standardizing vocabularies. It's about standardizing data structure, vocabularies, ETL conventions, data quality assessment, and standardized derived elements like phenotype definitions, which facilitate standardized analytics required for creating reliable evidence. The OHDSI community has produced lots and lots of scholarship evaluating the impact of vocabularies and the impact of ETL conventions on the reliability of the evidence generation process. Next. The idea of standardized analytic process isn't unique to OHDSI, so hopefully the idea depicted here shouldn't look brand new to others in this space. The main message is that standardizing data allows for standardized analytics, which produces standardized evidence devoid of heterogeneity from data structure, data content, or analytic implementation. This is something common to us all. We're all trying to improve healthcare decisions by for the sake of patients everywhere by leveraging the advantages of large multi-database studies. What differs are which databases are included and which analytic use cases are in scope for any given research question. Next. Again, I hope this cartoon is somewhat familiar. The OHDSI network studies rely on a common protocol and the OMOP CDM, although studies with combinations of CDMs are possible, however difficult to implement and interpret. But what's unique about this image is that OHDSI network studies accommodate different data types from different countries with different coding vocabularies stored in different database management systems, such that one standardized analytic package containing translatable parameterized SQL is executed across sites from which aggregated statistics compliant to local governance are shared with the coordinating site for evidence synthesis. Next. So these next papers support the need for 1, federated analysis, and 2, standardized transparent analytics. The first paper supports the need for network studies because if you execute the same study on different databases, you can get answers that can differ by up to an order of magnitude. The takeaway is that results are sensitive to database heterogeneity, so we can really only get closer to the truth by interrogating multiple sources. This second paper strongly supports the need for standardized and fully transparent analytics, whereby fully transparent, I mean using open source software and sharing one's analytic code. In this study, we found that if you give 9 groups of researchers the identical data, the identical tools, and the identical paragraph describing the exposure cohort definition from an excellent paper, the results can be wildly discordant. The team's cohort definitions built from the methods section paragraph plus code sets from the supplement varied from 1/3 to 10 times the cohort count reported in the paper. This type of implementation heterogeneity is unacceptable and seriously undermines reproducibility, which is a cornerstone of credible science. Next, please. Now, finally, I'll move to the main topic of this discussion, where I'll introduce one current approach to distributed analysis with a practical use case: research sprints that we refer to as study-a-thons. No one loves to hear that reliable evidence to support a high-stakes decision was needed yesterday, and I'm sure this type of pressure is familiar to all of us. An OHDSI solution to accelerate the research process is to bring together relevant stakeholders, including patient representatives, data holders, experts in clinical sciences, informatics, data engineering, epidemiology, statistics, and the ultimate evidence users to conduct the research in an open, transparent, short sprint lasting from 2 to 5 days. Well-conducted study-a-thons have rapidly produced reliable evidence where logistics, funding, and data governance requirements have been well planned in advance. This little heart emoji indicates that I love these events, but man, they are tiring. They can take a toll on you. But I'm introducing this approach because they're possible thanks to the OMOP CDM and standardized analytics, and of course, excellent leadership and tight project management. Next, please. So here's one example. After the COVID-19 pandemic shutdown, like most of the world, we quickly changed our plans from hosting in-person to virtual events. For 4 very long days in late March 2020, several hundred OHDSI scientists from around the world collaborated to rapidly design and execute research in support of the COVID-19 response. The short story is that this event produced a great deal of impactful research, which resulted in 12 published articles, 3 of which were highlighted in the NCEPT Guide on Method Standards in PharmEpi. I'll again reinforce that the OMOP CDM is a data model optimized to enable efficient analytics such that researchers can be quickly responsive to emergent needs like producing COVID-19 related evidence in weeks or regulatory responses at my day job in days. Next, please. So here's one of the example studies. Given that hydroxychloroquine and azithromycin had been endorsed as an effective COVID therapy by some influential people, despite shaky evidence, we sought to learn about the safety of this exposure combination from the experience of patients with rheumatoid arthritis, given COVID data were unavailable at the time. So we rapidly designed and conducted a large network study to assess the safety of hydroxychloroquine alone and in combination with azithromycin among patients with RA. Next, please. Our results were published on August 21st, 2020. We reported a short-term increased risk of cardiovascular mortality, chest pain or angina, and heart failure derived from hundreds of thousands of patients from 14 databases, which was highly concerning. Next. but this study's impact came before its publication. We posted a preprint on April 10th, which informed a cited EMA warning less than 2 weeks later, followed by a similar warnings by the FDA and Health Canada days later. And I'm very glad this combination therapy wasn't widely adopted across the world, since it's likely to have harmed a great number of people already harmed by COVID infection. Next. So this was all happening during the recovery randomized clinical trials when the recovery RCTs were being rolled out, arguably one of the most responsive set of pragmatic trials ever conducted. Among many findings, this trial reported on October 8th a numerical excess of cardiovascular deaths among hydroxychloroquine versus usual care among several thousand patients hospitalized with COVID-19 6 months after our observational study results. To be clear, we need to rely on trials like this. They should definitely be continued. But when decisions are needed immediately, real-world evidence supported by an efficient, scalable CDM can provide answers fast. Okay, next, please. So lastly, I'll reiterate what I began with. Observational data transformed to the OMOP CDM are of high fidelity to the source data, such that standardized, federated, international analytics can rapidly generate reliable evidence from across the world. And we can rely on this evidence to confidently make decisions that support patient health everywhere. Thank you.
Mike, should I just go ahead and get started? Or did you want to have any in between?
No,
Or
that's
Robert?
great. Thanks, Judy. Please proceed.
Sure.
Yeah, go ahead.
Hi, everyone. Thanks for having me. I feel like I've got a little bit of head start here because between the previous panel and then Jamie's talk, you guys have a lot of the background here, so I might be able to skip through some of these things that we've already talked about. But the bottom line is you've heard about the Sentinel Common Data Model at the last session, and then you've heard about the OMOP Common Data Model now, so I'll try to put it together for you. I think the differences have been sort of somewhat stated, but I'll try to illustrate them a little bit more. So go ahead to the next slide, please. So really briefly return to the Sentinel Common Data Model guiding principles. Like the OMOP Common Data Model, accommodates a wide variety of different types of data, including claims, electronic healthcare record data, registry data. And the main thing is the goal is to store that data at the most granular or raw level possible with the least amount of mapping, which is something you'll hear throughout this talk. Distinct data types are kept separate. So a prescription table is separated from a dispensing table. And at the point of performing any analysis, that's when all of those raw ingredients come together to sort of bake your cake, and make sure that you put together your outcome algorithm that you're looking for. We always really rely on local expertise whenever possible to understand more about the data. So as Mike and Robert know well, I have talked to them at length about how Canadian data is different from American data and learned a lot about it. And we take that local expertise into account whenever we think about doing any type of analysis. But ultimately, the Sentinel Common Data Model, the main consumer of it, or the first user of it, is the US FDA, who uses it for regulatory decision-making. And they prioritize analytic flexibility, transparency, and control. Next slide, please. So this is just a brief graphic of the Common Data Model. You can see up to the left, you have administrative data. So this is things like enrollment and various medical product utilization. We have some specialty tables. So there's a mother-infant linkage table. There's a table specifically feature engineering for natural language processing based ideas. There's some registry information. We have death registries and cause of death registries in the US, clinical data, and then patient-reported measures. Next slide, please. So this is, I think, a paper that Mike talked about last time you guys met, but just to reiterate, this is by Sebastian Schneeweiss. It's sort of this nice primer on what are the different common data models, what do they do, right? And just to take a, sort of brief reminder. So they're at the bottom in that raw data structure that you see there. That's all that source data that everybody has at individual, sites. And then one type of a data model, an organizing common data model, takes all of that and essentially tries to keep it as close to source as possible, but creates standardized fields so that you can leverage each of those different source data sets in the same way because they've been structured into the same standardized field. Now, then there are the rules layer. And so let's think about that as the analytic layer where the sense making happens or where the knowledge generation happens. And so in the OMOP common data model, a lot of that happens already embedded in the data model because the vocabularies that that Jamie talked about have been created, and then that information is then placed inside the data model itself. In the Sentinel data model, there is instead a structure where at the point of each study, that's done each time using what's called an adaptive rule system, where we have a main structure with input file driven processes. And then of course, there's the more traditional study, which sort of Jamie led with, which was the old way of doing federated analysis, which is each individual site does line programming for that particular study. And so this is just, again, another way of organizing how these different things fit into the same ecosystem. Next slide, please. So what's the big difference? We've talked about it a little bit, before, but to just state the main difference between an organizing common data model and a mapping common data model is the use of this mapping, and then the placement of that mapping in the data model itself. So this is just a picture illustrating the mapping and illustrating particularly in the OMOP common data model, there are two standards. There is the SNOMED standard and the RX norm standard, where everything is mapped to those two standards. So next slide, please. And this is a table Jamie also showed, so we can skip over this really fast. The important point here is just to note that most of the data is mapped. So 11% of these fields are source data fields. The majority of it is mapped. Next slide. So these are some questions, and I encourage you to go to the frequently asked questions site that OHDSI and the OMOP community maintain, where they talk about sort of their philosophies and the things that they do for various reasons. But the thing that I, wanted to sort of point out here, and we've talked about before, what are the big differences? And one of the big differences, one of the things, most of the time you use a data model so that you can use a standardized toolkit. And then the question is, where do those standardized toolkits work against? And so here, I've underlined some important points for you, but I think, not speaking for the OHDSI community, but allowing them to speak for themselves in their frequently asked questions, their feeling is the use of the source values fails to take advantage of the benefits of the common data model. And that's where I have to say that I respectfully disagree. And so I think that the thing that, before we transition, sort of thinking about why would the source data be so important? Why does she keep harping on this? Why is the source data something that we should care about so much? So this was created for the Food and Drug Administration. And the Food and Drug Administration, just to think about the type of data it normally uses, more than 90%, probably a super majority of data, come in the form of pre-licensure data that supports understanding whether a drug is safe and efficacious in order to be used and licensed by the government. And so most of that data is primary use data. So these are data that have been directly collected for the explicit reason of doing this study. And every single data element has been planned, and they have decided how they want to measure it, and then they have gone out and they have measured it. And all the data we're talking about here is secondary use data. So automatically, it's not the same as when I've gone out and I said, this is exactly what I want to measure, and this is how I'm going to measure it. So we're taking data that's already been measured for a different reason, and we're trying to make sure that we can use it in a comparable way. Now, there are many people at the FDA that will say it will never compare to primary data. So what we typically are trying to do is get as close as possible to what that primary data looks like. And that just means that we try to leave that data as raw as possible, so we do the least amount of manipulation of it before we move on. So the next slide here is going to show you some guidances that the Food and Drug Administration put out. So the first guidance in the use of real world data, and there's been a lot of guidances recently that the FDA has put out about these data, because there's a lot of intent to potentially use these data in pre-licensure applications as opposed to just in the post-market where they've traditionally been used. And so I just want to highlight for you, again, guidance for industry, but guidance that I live by because I work under the auspices of the Sentinel system, that researchers should ensure that curation and transformation processes do not alter the meaning of the data or cause the loss of important contextual information. And then there's some other supporting comments in there. The next guidance that I'm going to show you on the next slide relates to the use of those data in pre-licensure applications, where the FDA should be able to interrogate those data for their integrity. And so there's some highlights there, but to ensure that relevant patient level data can be provided to FDA and that the source data, which is defined as all of the original records and all of the clinical findings and observations and other activities that it needs in order to verify that that real world data is appropriate for the use that it's being put forth for. So next slide. So what does that all mean? It means that the Sentinel system uses source data to the greatest extent possible to maximize transparency and analytic control by the investigator and to minimize any introduced heterogeneity that can result from mapping. So for our purposes, embedding or map concepts in the data model is not what we believe is required for multi-jurisdictional research. It may be a convenience for these purposes, but it is not required. Rather, the preference is to work with local data owners. And by that, I mean people like Mike, in Ontario, people like all you folks in Canada, to understand how those codes work, how those concepts work, to understand that we can then use that data together in a way that appreciates the heterogeneity that may naturally exist there without introducing any undesired heterogeneity in exchange for the convenience of mapping. So next slide, please. So this is a slide about essentially the concept of the data model being parsimonious. We only keep the data we absolutely need. A lot of this is also related to size considerations. So if you have to store both the source data and the map data, which OMOP does, you have more space that you have to use. You basically have more size and it takes up more room. For the databases that we use to support the Sentinel system, the FDA pays for the maintenance of all of those databases. And so essentially we try to keep the most parsimonious footprint possible for cost considerations, in addition to storage and other sort of processing pieces. Next slide, please. So let's talk about an example. Say I want to measure BMI. BMI is a super important covariate in so many different studies. So our intent is to keep it as raw as possible. So we have a clinical table where we collect height and weight that's dated. But we also could use diagnostic codes if we wanted to measure obesity. When we would do that, we would always designate them separately. We'd say, this is a diagnostic code for obesity. These are clinical measurements. Next slide, please. This is an older slide, but it's a graphic with Patrick Ryan giving some advice to folks. For folks who are not familiar, Patrick Ryan, I think of him as like the godfather of OMOP. And so essentially, he's telling folks, hey, when you're mapping this, it's okay to map an ICD code or a measurement to the same concept. This is the map concept to use in the data model. Next slide, please. So this is the basic difference. We're very strict. The measurement goes in one place. It's the clinical measurement. The diagnostic code goes in a different place. If you later want to combine those analytically, it's up to the user to do that, but we're not going to do that in the model itself and embed it there. We think that map concept is potentially a loss of granularity that we don't want to have when we go to use that data in a study. Next slide. So I've gotten to know Canadian data quite a bit over the course of the last year. And one of the things that I really want to caution about with mapping, because I know that this group is interested in adopting the OMOP Common Data Model, is to really be careful around the granularity associated with the setting. So my understanding is that right now, ICD-10 Canadian modification is used in inpatient and emergency department settings to talk about concepts, clinical concepts. And that has up to 6 digits. And so it has quite a lot of codes, quite a lot of granularity. But in the outpatient setting, you guys are only using ICD-8 or 9, which sometimes is truncated to 3 digits at the most. And so this is the issue of where does this mapping create problems? It's when you take something that is, too general, and then you make it more specific than it is, misrepresenting what it is, or you take something too specific and you lose the specificity because you're forced to map it into something more general. Both of those things are problems. And so I think here with, Canadian data and the particular, peculiarities of it in the fact that the inpatient emergency department setting is functionally just incredibly more granular than the outpatient setting, real care has to be taken to be able to use these codes effectively to actually capture, what you're trying to capture. So consider diabetes. In the ICD-9, 8, or 10 framework, there's only 3 digits that describe the ICD, the code for diabetes, but that doesn't differentiate between type 1 and type 2. However, if I'm in the ICD-10 setting, I can capture 6 digits. So I can actually capture that granularity. And I want to make sure I understand how to use those things together, because in the end, all of our goal is to prevent mistaken interpretations that might, arise that could come out of something like a mismapped value. So next slide, please. So this is just a cautionary tale to say, even when we do our absolute best to try to minimize what we can with respect to heterogeneity, and we use, local expertise, we have people who really understand the data, define all the concepts, use things that have been validated, we're already challenged by that. And then if we, the next slide here is sort of saying, well, let's keep that at the point where we are looking at heterogeneity without introducing anything additional in that point. And this is a study that we did together with Canada with CNODES. In this case, we used the Sentinel Common Data Model. We left the Canadian data as Canadian data. We did not map it to any other concepts, and we still had a lot of heterogeneity. And so, it's just a cautionary tale to say, we want to make sure we're doing our very best with the data we have and not misrepresenting anything. Next slide. So final thoughts, to embed or not to embed a map concept. Why might I want to? I can get to it a lot faster. Jamie demonstrated that by talking about the COVID-19 studies that they did and how quickly they were able to get to those concepts, right? It's kind of convenient. I don't really have to know very much. I can just, pick the concept I want. I don't have to spend that time really connecting and learning about that source data in many, many different jurisdictions. It's really great for settled definitions, things that don't change over time, and they don't change depending on your research question. And they don't need to have more specific or more sensitive or other types of variations that are there. So the disadvantage of embedding that map concept is, again, you potentially lack that flexibility to adapt it to your research question as you need to. There's significant front-end work involved in mapping an entire database of codes. And I think actually my understanding is Canada is experiencing this now in trying to adopt the OMOP Common Data Model. Mapping is a challenge, and doing it all up front for a whole database instead of a cohort by cohort strategy, it's quite a lot of work. And then of course, there's the actual physical size and footprint of the storage of those data concepts. So I think we could all agree that we would be fine to map when there's no harm that mapping changes the interpretation of what is being measured and would cause erroneous conclusions or interpretations as a result of that mapping. And with that, I will leave you with the last slide here, and you can click this because there's an animation here. So this is one of my very best friends, Josh Gagne, who now happens to also be the head of epidemiology at J&J. He wrote this paper a few years ago. And this is a commentary paper on an original research paper. The original research paper I left in the slides, but didn't go through it for the purposes of time. The original research paper shows that the same study performed in OMOP and Sentinel Common Data Model produced different results. And so Josh was asked to give a commentary on this. And essentially, he leaves us with the last thought, which is, to the extent that common data models facilitate scaling of the most rigorous design and analysis plans, then bigger will be better. But scaling of inappropriate design and analysis methods will lead to results that are more precisely wrong. And of course, that's what we all want to avoid. And so with that, I'm happy to turn it back over to Mike and Robert, and thank you for your time.
Thanks, Judy. Thanks, Jamie. Thanks, both of you, for this. This has been a very thoughtful session. I'm going to invite questions from the chat. There was one that came in while you were speaking, James. I'll just read it out. Thanks for sharing the study-a-thon model. Do you know what the lead time was from when the event was conceived to when data access was granted, i.e. how long did the governance process take?
Many of the participants were aware several weeks in advance of the planning, which gave adequate time to get permission to use the data for this purpose. But it definitely varied by participant. Some participants at academic institutions took longer than private organizations that had more readily accessible data. But there was definitely a lead-in requirement for people to participate, which is why I said that the limitations of the study-a-thon model, it's conditional on getting a lot of upfront logistics and governance out of the way. And of course, it's logistically challenging to get people from all over the world into the same room for 5 days. Like, that's just hard.
Probably was easier when it was the same Zoom room than the same physical room, I would imagine.
There's a magic that happens when you're all in the same physical room. And
True,
not the magic
true.
of transmitting COVID.
Okay, well, here we go. The next question, this is for Judy. If Sentinel doesn't map ICD diagnostic codes to a common standard, e.g. SNOMED, how do you deal with different ICD versions being used in different jurisdictions?
So what we do, so for example, in that study that you were that we captured here, we use a common analytic package, so the same program, but in the input file, we have a particular combination of the table in which the code is found, the code itself, and then the coding class, if you would. And so in the US, we use ICD-10 CM codes. So it's designated as an ICD-10 CM code with the code that's being used. In Canada, they use ICD-10 Canada codes. So we designate that as a Canada code with that code. So essentially, every code has associated with it a designation, and just in your overall outcome algorithm, each of those types are laid out and then included in that concept. So instead of the mapping, you just take, again, the advice of the people locally, so in this case, Canada, how would you do this? And they look at these codes and they put it together, and then we just stack them. It's just a vertical concatenation. So do you adapt the protocol locally? We don't adapt the protocol locally. We work with the local jurisdictions to have the common protocol before we start with the common coding. So, and when I say common coding, I mean the Canadian coding and the American coding, they get stacked together for the same concept. I'll read the other one. How does the Sentinel model handle textual information containing clinical notes? So all contextual information and clinical notes, we use a table that specifically designates that this is what we call an engineered feature. So this uses an NLP pipeline. The pipeline is labeled and the pipeline is version controlled, and there's a bunch of information about it. And when that pipeline is applied to a database and a concept comes out, for example, smoking, it has a label and it has a designation of it, and it's indicated that it's an engineered feature, meaning it's come from the notes. It is not inherent in the source system with a structured piece of information.
Thanks. There's a good question that's a big question that I had in my notes as well that I wanted to ask both of you, which is, how is heterogeneity explored? And are there different tests and ranges for agreed unacceptable to acceptable heterogeneity? The questioner is modeling this on sort of a systematic reviews framework. I had the same question, but at the level of the database. In Canada, when we're looking at our administrative claims data, the structures and the populations are fairly common across the country. I know in both the Sentinel CDM and in the OMOP studies, you work with much more heterogeneous data source types. So I'd be interested in both of you commenting on that.
I can start in that, this isn't even this isn't specific to any CDM. Just like heterogeneity of results is a mixed bag of things. It's a mixed bag of truth and various sorts of error, and untangling that is no easy task. So you can, of course, measure heterogeneity through traditional methods like I-square or tau-square statistics, or just visually inspect it, but really drawing out where that heterogeneity comes from is a challenge. And I think we need to accept and learn from heterogeneity that is because of actual population composition differences, and try to suss out what is actually site-specific biases, like measurement errors in one database compared to another, which is the basis of, different health systems collecting information in different ways. So like simply put, measuring like incidence or effect estimates across databases, you can do that mathematically with I-square or tau-square statistics, and then make decisions on whether or not it is appropriate to synthesize that evidence across sources. But it's subject to the constraint or the limitation that different data sources will have different errors than each other. And so there needs to be that thoughtful approach to before synthesizing any evidence, knowing that it's a mixed bag of truth and error.
I'm not sure I have anything
Judy,
more
any
to add to that, Robert. I think that covers it really. I mean, in general, we always do a data partner specific set. So we look at whether there are outliers, or we do always an inspection of it. We rarely go to the level of statistical testing to look at differences, but we also have expectations. We have, for example, commercially insured patients. We have publicly insured patients, either through in the US it's Medicare and Medicaid, but they're equivalents in Canada as well. And so we have certain expectations going in of where we're going to see differences that, it's not. What gets surprising to us is if patient populations that are largely similar then have differences. And then, there are differences in formularies and things like that that can give rise to some of these things.
I see another question that was from Colleen that asks about other direct head-to-head comparisons between the two approaches. And this just makes me want to go back a second to Josh's commentary on the Shu paper back both from 2015. So like Josh's comment where he speaks about inappropriate design and analysis methods, that doesn't actually refer to the CDM design. That refers to the study design that is creating an effect estimate. And what Shu and company reported was that they showed minimal impact from conceptual differences on identifying safety associations in the CDMs, but where they did see heterogeneity, it was on the basis of using different methods in the two different ecosystems. So that like inappropriate design and analysis methods as being a problem, that's of a study design method, not a CDM design. So like I reread those papers earlier today, and I just wanted to point that out, and I recommend you all take a look at both the Shu paper and the commentary.
This next question is a interesting one. Has heterogeneity been explored at a patient-doctor level by OMOP or Sentinel? As a clinician, I feel I'm already adapting my diagnosis and treatment vocabulary to ICD-10 and CPT coding, which is already a lossy and inaccurate mapping, and things are chosen sometimes due to being close enough. Have either of you gone down to that sort of granular level of consideration? I'm trying to think of what one could do to assess that, but
I think for the most part, in my mind, that's about the practice of medicine in the U.S. And certainly the FDA is very reluctant to sort of wade into those areas with respect to the practice of medicine. Really, we're trying to make use of already structured data for its sample size. So there are places, for example, in using an NLP engine that looks at various unstructured clinical texts and tries to make meaning out of that. But that might not be making the right meaning. So just it's one of these things that I think is not necessarily an easy to solve problem. I appreciate the feeling of that certain types of the coding are still not getting at what you're getting at. Certainly, I think the being forced in the outpatient Canadian setting to only use the 3 digits of the ICD system is tough. So I certainly sympathize on that front.
I feel like this is hinting a little bit towards phenotype algorithm validation, studies, and the idea that, you know, as a clinician, you're putting in some reimbursement code, intending to capture something as complex as what's happening physiologically inside a body. Like there's, you know, just that game of telephone creates error. So again, regardless of the data model being used, this type of idea of validating not only some code or another, but the combination of code sets plus temporal logic to create a proper, like, computable phenotype approximation of a clinical condition definitely requires a great deal of work. And that is consistent across anyone in the real world evidence community, and trying to come up with ways that are more efficient than locking clinicians in a room with a stack of charts in order to validate it is, you know, a huge opportunity space for us. And there are several proposed informatics solutions to this at the moment, but you know, they're going to need a little more research and socialization into the community before they're widely adopted.
Yeah, I think this is just a really hard problem in terms of, like Jamie said, you almost need to lock a bunch of people in a room with a bunch of charts to do this kind of exercise really, really well, especially if you're getting down to the granular level of actual ICD codes. But it's something I think that all of us need to be aware of when we're working with these sorts of data. Okay.
So I hope I haven't misinterpreted the question though. Like, Eric, did I interpret your question correctly?
I think so, but
okay.
So we have time for one or two more questions, which if I don't see any in the chat in the second. Okay, here's one from Mahmoud. OMOP is known as a patient-centric clinical data model as opposed to a visit-centric model. Judy, how do you conceive of Sentinel in this?
In Sentinel, I mean, the patient ID is the unique key across all of the data sets. So everything relates back to the patient. So yes, there are visits that are captured as encounters. For example, you go to an inpatient, you get admitted to the hospital, you're in a stay, you have an emergency department visit, it's coded as an emergency department visit, you have an ambulatory visit. But all of it, ultimately, it relates back to a patient, and the unique key amongst all the tables is the patient. So I don't see it as much different.
I agree with that, and that OMOP is indeed a patient-centric model, but the data that it relies on is encounter-based longitudinal data with the unique key per patient. So it is indeed both. And I do believe that much of the, you know, the Sentinel CDM source data are similar to many of the OMOP CDM data as well. Like they both are from the same, many of them are from the same sources, which are, again, these person-centric encounter-based longitudinal databases. And I do believe the models are similar in that they capture both of that with high fidelity.
Okay. Alexander, do we have time for one more question?
Yes, I think we have time for one more.
Okay. If in that case, I'm going to take the moderator's prerogative and ask one about software. Jamie, you mentioned a couple times OMOP is based on an open source platform, and I'm to my knowledge, the analytic software is in R. Judy, to my knowledge, the analytic software for Sentinel is built in SAS. Those two have different trade-offs in terms of R is much more flexible and suitable for user-initiated code. On the other hand, package dependencies become a bigger problem with R. SAS can be more cumbersome and limiting in terms of what you can do with it, but if you're working with a consistent SAS version, you usually don't have to worry about package dependencies. Wonder if either of you had any thoughts on this sort of the general software issue and maybe where analysts should be investing their time.
So there is a large open source software development community within OHDSI that produces a huge number of software packages, many in R, but many in other languages as well. A lot of this relies on SQL in order to basically access the data in a relational database, but much of the high computation, like highly compute intensive routines run in many of these statistical packages run in C++ or Java or all sorts of other things. So like OHDSI is not an R shop. OHDSI does rely on R in the sense that many people can contribute to that, but it is not like, it's not some one of the principles of OHDSI is not to use R. It is just that is a convenience. Unlike mapping is not a convenience. Mapping is a huge undertaking in order to enable international collaboration, and whereas like R is a convenience, but it's not doesn't rely on it. But the open sourceness of it is actually what is most important, in that it would indeed entail a lot of investigator burden or reviewer burden to go and look through all of the code in one of these great big long multinational or international studies. But that type of burden is, not worth the trade-off of compromised scientific integrity. Like, it should be all there and it should be all auditable. And the time it would take to actually see from start to finish what has happened is, worth it. And we've seen, as I showed in this reproducible variability study, where all you have is a set of codes in an appendix and a paragraph writing about how to define an exposure cohort, just how wrong that can be. So it's why the main point in software in OHDSI is not so much which language it's programmed it in, but it's whether it's available for others to see and assess. And we encourage that. That's why it's there.
Thanks, Judy. Any comments on this?
Just that the SAS code that's used is published, and everybody can look at it and use it. There is a burden of the licensing associated with SAS. There is some, I think, free academic licenses, but the main reason that SAS is utilized is because the largest data sets that we have in the US, particularly the Medicare and the Medicaid data sets, essentially the shop that maintains them is primarily a SAS shop. And so being able to use that data effectively in some ways necessitates the use of SAS. If people want to take SAS data sets and export them into, CSV files or, other stuff, people are welcome to do that sort of thing. But yes, for the most part, it's in some senses, it is a necessity of the underlying data source systems that are the most important to the FDA for their analyses.
Can a user export the SAS data files that have the US Medicaid and Medicare data in it to try it themselves?
They can export the input files. They can't export actual, like the Medicare and Medicaid data is under data user agreements.
Right.
So but the package itself, if they have data formatted in the common data model, anybody can take a package and run it against there.
I see. Thank you.
Can share tools, not
Great.
data.
So thanks very much to both of you for a great discussion.